Okay, we're going to be starting on a new topic, which is digestive system. We're going to be covering anatomy first, as we often do, and then we'll get into physiological processes as we go through. So let's go to the lecture notes and get going. So this very first picture here, you can find in your book on pages 869, and I actually brought up the book filled in a very similar version, and I'm not going to fill it in. I think you have something in your lab this week that's very similar, so you can go ahead and use this picture, the one in your lab book, to fill these in. But basically the thing we want to look at is that when we're looking at this is we're looking at the digestive system, or what we call the alimentary canal, or the gastrointestinal tract, which is sometimes abbreviated as the GI tract. And it's a continuous tube that starts here at the oral cavity, goes down through the esophagus, the pharynx and esophagus, into the stomach, and then it goes through the different parts of the small intestine, leaving and then going up, over, down, and around and out the large intestine and out the anus. In a cadaver, it's around 30 feet long, a little shorter because of contractions and the way the tissue is in us, but that's it. And so we're going to go through all of these different parts over the course of the next series of lectures. So you'll get to know all of those later. But I'm not going to fill this in. I'm just going to write that it's on page 869, and you can go ahead and take a look at it as you need to. Okay? One thing you'll notice is that also on here are things that are called accessory structures. And accessory structures are structures that are either outside the digestive tract and are secreting into it, or they start out the digestive tract and they protrude into it. So that's kind of important to think about, that we have certain structures that do that. And so these guys, and I won't underline all of them, but like your teeth, your tongue, your glands, salivary glands, your pancreas is secreting enzymes. They lie outside the digestive tract and either protrude or secrete into it. Okay? Now, what are the digestive processes? There's a picture here at the bottom of the page, and it can be found on page 870. And page 870. And let's switch over and take a look at that, so you can see it in a little bit better detail here. It's a little bit larger. So what this shows is all of the different processes that are occurring in the digestive system. And some of these processes occur in multiple areas. Some processes only occur in one area. So we're going to go through them all, and we can just kind of follow along in these colored areas here, and we can look at them and then note what is in the lecture notes. So let's go ahead and do that. So the first one here is ingestion, or eating. And I think most of us understand that one pretty well, taking food into the body. No trouble there. And then the second one is a set of different things, but the overall word is propulsion, which is motility or movement of food. Okay? Now, there are two main ways that we move food. We can swallow, and we can have peristalsis, right? Swallowing is voluntary. We choose to swallow something or not. Peristalsis is involuntary. And what peristalsis is are alternating waves of that are of the smooth muscle inside our canal that squeeze around the food and push it generally distally through the tract. Now, one thing to note, we get to use proximal and distal here. I'm going to go back. So this is the proximal end of the digestive system where food starts. And as we go through, we get to the end of it, and we call this the distal end. So generally speaking, food will move through and go out the distal end. So that's what we're referring to there. Okay? Now, there is a picture of peristalsis on page 851, and we also have, I think, a picture I thought, or maybe we don't, in the text or in the notes, but let's go find it really quickly here. There it is. So peristalsis, notice what you're seeing here. You're seeing what we call a bolus of food. So a bit of food that we chewed, we put into like a little ball in the back of our mouth and we swallowed it. And now it's called a bolus of food, and it's going down our alimentary canal. So this could be somewhere like the esophagus, for example, or the small intestine. And this food here is, as it enters in, it kind of stretches out the canal, and what happens is then contraction occurs right behind it. And when contraction occurs behind it, it pushes it this direction. And then we continue that contraction and continue pushing that way. So it's kind of similar to like if you were to take a tube of toothpaste and you took your finger and you started to contract it, you unscrewed the top of the toothpaste and you squeezed, you would push the toothpaste out the hole, the opening of the toothpaste, and you could run your finger all the way behind the toothpaste and push the toothpaste all the way out if you so chose. That's peristalsis, okay? Now it's involuntary in that it just happens because there's food there and then the smooth muscle reacts by contracting around it. So motility, as we just said, is activity of the smooth muscle within the organ wall, right? And it must be regulated carefully because if the food moves through too rapidly, there won't be enough time for food to be digested or absorbed. So if you think about it, what we're doing with food is we're putting it into our body, and then we are breaking the food down into usable molecules that we can absorb into our bloodstream. Now, if we're moving food too fast through our system, the food will not get broken down and absorbed correctly and will pass through our body partially undigested. So we want the motility to go slow enough so we can extract nutrients, but fast enough to move things along and to allow us to eat more food later. All right, so we want careful regulation. We want because if the food moves too rapidly, there'll be not enough time for the food to be digested or absorbed. Now, mechanical digestion is breakdown of food into smaller pieces. There's 3 different ways that we can do this mechanical digestion: mastication, churning, and segmentation. Okay. Now, in each of these cases, the food is not moving from its location, but it is being broken down into smaller pieces. Okay, so the first one, mastication, occurs in the mouth, right? And this is just chewing, cutting, smashing, and grinding. If you want to write chewing next to it, you can do so. We're taking a big thing of food and crushing it into smaller pieces. We're also mixing it with saliva or liquid. Then, when it reaches our stomach, we do something called churning. And this is where the stomach's smooth muscle will smush and contract and break the food and smash the food even more, separating it and kind of liquefying it more. Then we have the small intestine segmentation. Now, segmentation is a localized contraction that separates food, breaks it apart, but it also helps mix the food with digestive juices. And that's good because now the enzymatic juices that are going to break the food down are getting very well mixed in with these smaller pieces of food. Smaller pieces of food means bigger surface area. And if we have a greater surface area, it'll be easier for the next step, chemical digestion. We have a picture of this segmentation right here next to peristalsis. So here's our bolus of food here, and here is some, say, digestive juices like pancreatic enzymes. What we do is we do contractions in place. We're not moving food down. We're just smushing the contractions. So this would be like if you were just holding on. Let's say you closed up that tube of toothpaste and you just started pushing your fingers back and forth in the same spot. You'd be mixing the toothpaste in together with each other. So what happens is this food and these enzymes get kind of separated and mixed, and eventually as they keep getting mixed, they become incorporated, all right? And we now have a mixture of the enzymes and the food. that makes chemical digestion our next possible thing. Chemical digestion occurs mainly in the stomach and the small intestine, you can see here. And this is chemical breakdown of macromolecules into their monomers. Now the macromolecules are right here, and this should be review from Intro Bio. Carbohydrates are the macromolecules, or we sometimes call them polysaccharides. Proteins are macromolecules, and lipids are macromolecules. Monomers, what we break them down to, are the single subunits, monosaccharides, amino acids, and fatty acids, and glycerol or monoglyceride. That's what we want to get eventually. And so we're going to use chemicals, mostly enzymes, to go ahead and release. And so once we get to the monomeric form, once we get down to the monomers or very close to it, we can absorb them into the bloodstream. We cannot absorb the whole carbohydrates, the whole proteins, the whole fats into our blood. We have to break them down into their monomer subunits. So again, the other thing about timing is important is that these secretions, these hormones and enzymes, have to be regulated and released at an appropriate time. Otherwise, we might not break the food down properly. What if we put food in our small intestine and then we didn't release enzymes until 20 or 30 minutes later? The food may have moved along the small intestine and now we dump the enzymes in and there's nothing for them to digest. So it's important that this is a very regulated system. All right, so now we're getting to absorption, okay? Absorption. Now, absorption is a passage of the food from the GI tract lumen. Remember, lumens are openings. So from the inside of the GI tract to the blood, and in some cases, the lymph, all right? And that's what we're going to be doing next, okay? So we're going to pass the food from the GI tract lumen to blood and lymph. Okay? So that means that the products must go through the mucosal cells of the GI tract. We have to go into the mucosal cell and then into the blood. Absorption is not regulated. So everything else had been regulated up till now. So whatever you eat might be absorbed. So the you are what you eat is kind of correct. Once it goes into your body, if it can go into your bloodstream, it will. If you have a gigantic piece of birthday cake and you're like, oh my gosh, I ate way too much sugar, unfortunately, once it gets into your digestive system and starts getting absorbed, it's going to get absorbed. So absorption is not regulated. Now, the last thing is defecation. Now, defecation is mostly the elimination of indigestible or non-absorbed substances from the body. There are a few things that we put into our fecal matter, like bilirubin, for example, which gets converted into stercobilin, that brown pigment, if you remember that from Biology 232. But for the most part, anything that we can't ingest or we can't break down will go out in our fecal matter. Okay, so that's our last spot. All right, so we want to go over some basic functional concepts of the digestive system before we go any further. So because we have a lot of regulation, we need to have ways to control the movement and the secretion. And we want everything to work together so we have optimal conditions in the whole GI tract. Okay? And there's a local neuronal control of the GI tract, and this is due to this local neuronal control, and it's called the enteric nervous system. And the enteric nervous system is a subdivision of the PNS, peripheral nervous system. Okay? And two aspects of the regulatory mechanisms that we want to talk about here is that most digestive reflexes are initiated by sensors, right? Receptors that we have in the GI tract walls. So think of kinds of sensors we might notice. Mechanoreceptors, those are pressure receptors. So if you put food in an area, you will feel it against the wall of the GI tract. Osmolar receptors. Okay, osmoreceptors are water or liquid receptors, right? How many dissolved solutes are in that water? And then chemoreceptors, looking for specific chemicals. Maybe it's sodium, maybe it's sugar, maybe it's pH. These things, these receptors will stimulate the activation or inhibition of secretions and motility. So if we notice something's there, we will release what we need to handle it. If we have a lot of protein, then we should probably release a lot of enzymes that can break down protein, right? So these sensors are activated by different things. So the mechano ones, like this is just a review of what I just said, but I'll write it in here. Let's put it up a little higher. The mechano ones stretch of the GI wall, right? The osmolarity or pH levels of contents are the osmoreceptors, and the presence of substrates and different chemicals in foods are the chemoreceptors. Okay. Now, these stimuli don't just activate nervous system, they can activate hormonal pathways also. So we can get both nervous and hormonal pathways. And what we end up doing then is when we stimulate these, what are we going to do? Well, we're going to use our effectors, right? Because this is like a feedback loop. We notice something's there, our brain decides what to do about it, or our gut decides what to do about it. And it sends message out to effectors. And so these are our effectors here. Smooth muscle, right? Muscles and glands are effectors. So we might break things down mechanically by using our smooth muscle, or we might start peristalsis to move things along. We also have glandular secretion. We could secrete digestive juices, which is exocrine, like from our pancreas, or we could secrete endocrine secretions, hormones, like we were just talking about in the endocrine section. So we'll come back to some of those things a little bit later, the hormones, that is. But let's look at the this whole GI tract control neurally first. Okay, so we're going to look at neural control first. Here, so we're going to do neural control first, right? So there are two types, intrinsic and extrinsic. Okay, intrinsic literally means inside the GI tract, where extrinsic means we're going to send messages out of the GI tract and direct and up to the brain or spinal cord. Okay, so let's look at some of these. And by the way, this picture here is on page 875. And this picture is similar. It's not the exact same one, but it's similar. Page 875. And so I think it's fine to use this one if I can type everything in. Maybe I'll have some trouble typing in or putting some things in, and you'll want to do it later. But so the first thing is, let's start with intrinsic things that are happening in the brain. These are called short reflexes. And the reason why is the stimulus is happening inside the GI tract. So let's say we put some food somewhere and we stretch some receptors. Okay. And so there's different receptor types. The different receptor types that we just talked about. Okay. Different receptor types, okay? And these go along what we call a local neural plexus. Okay, a local neural plexus. Let's see if I can move it over one. Is that okay? Does that not bother you? Okay. Local neural plexus. Now, that local neural plexus is just the enteric plexus we just talked about. Short reflexes, enteric plexus. And we then send messages directly to effectors. And if you remember what effectors are, They are, I don't know if I can write them in here, muscles and glands, right, are effectors. Okay. And then we have some response. And what's the response that we can do? We can either, we already talked about this, we can contract or we can secrete. So we can either do some muscle movement, the muscles will contract, or the glands will secrete. Okay? So we sometimes call this the gut brain, the enteric plexus, because it's doing a reflex, right? This is like a reflex. And it's not using our normal control centers, our brain or spinal cord. It just does everything inside the gut. It notices there's something there, it sends a message locally, and it releases whatever it needs to release or contracts. Okay, so we get local hormone release or local contraction, right, from the GI tract itself. Now, we can have extrinsic pathways as well, and these are long reflexes. And these can be mediated by stimuli inside and or outside the GI tract. And it involves the CNS. So this one does involve the CNS. And you can see these long reflexes here. So let's say a receptor, a mechanoreceptor, picks up that you put something in your small intestine. You move some food to your small intestine. It will start to do this plexus, but it'll also go up here and activate, I can't write it in the right place, the CNS, our brain and spinal cord, and it will then send to make decisions like a normal feedback loop and send messages down efferently to the local neural plexus, to muscles and glands, and we would do something. We can also get external stimuli, and that's what this meant when I underlined here, mediated by stimuli from inside and or outside. So what kind of outside stimuli can we get outside of the body that might start stimulating this? Well, the sight, the smell, the taste, the thought of food. All of those things are external and send messages to our control centers and then can do things. So think about maybe going to a movie theater, and the moment you walk in, what do you experience? The smell of popcorn, the sight of the popcorn machines popping and all the bright colors and with all the advertised products. Now you're thinking about, oh, I'm going to get a big thing of popcorn for this movie. All of those things are going to start your digestive system doing stuff. It will start moving things down the line to get ready for this popcorn you're about to eat. It will start causing contractions and constrictions. If you're waiting in line and your stomach is growling, what that growling is really the stomach contracting. So, and even the moment you taste the food, we send messages down to the digestive system to tell it to get ready for food is coming. So those are external sources that are important. Okay. Let's see. We will continue on here and just do the next couple pages. We'll see how far we can get. But we want to talk about the relationship of the digestive organs and the peritoneal cavity. So if you remember, the peritoneum was the largest serous membrane of the body. And this is where all of the abdominal cavity organs were, right? And we have a parietal peritoneum and we have a visceral peritoneum, right? I'm going to put some abbreviations, even though I can type in here, it's hard to put everything in. So what I'm going to do is for parietal peritoneum, I'm just going to put PP, okay? And for visceral peritoneum, I'm going to put VP, okay? And I'm going to write these down where they are. So parietal peritoneum lines the cavity walls. And right here in this picture, and again, by the way, this picture does show up in your notes. Farther in the chapter here, let me pull it up really quickly. I always forget exactly where it is. It used to be on page 889, but I think they moved it farther. Oh geez, I can't find it. There it is, on page 908. Page 908. Page 908. 08. Okay, and all right, so down at the bottom here, you can see a couple spots where we can look at the parietal peritoneum and the visceral peritoneum. So the parietal peritoneum goes around the outside edge, which is right there, and then right above it here, you'll notice that the visceral peritoneum is going to then cover the organs. And in this case, it looks like it's showing us covering some of the small intestine parts. Okay, so the whole lining, the whole parietal peritoneum goes, here I'll use my little pointer here, right here, goes all the way down and around, and then it doubles back and now it becomes visceral peritoneum for all of these things. And it does that in all the organs, okay? Now, you'll notice that some of these organs are hanging or being held by little lines and ligaments, and that's what we want to look at next. Before we get there, though, remember this kind of purpley part, that's the peritoneal cavity, and this is the serous fluid. This is where the fluid is, and it helps reduce friction, so the organs slide against each other without causing any damage. Now, the mesentery is in general, so this is a definition here. So let's call this a definition. Okay. 2 layers of serous membranes with loose connective tissue between them. That's mesentery. Now, many organs are being held by mesentery from the abdominal wall, and you can kind of see this. See, there's a line right here, and there's a line right here that goes down. Those are holding the organs kind of in place in space in the peritoneum. Right, so that's one of its jobs, and you can see that in lab. If you look at the human torso model, you can see the mesentery sometimes. If you turn kind of some of the organs out, pull them out of the cavity, and you can look and see them there. All right, and these also are passageways, the mesentery, for blood vessels, lymph vessels, and nerves. So they can go through these areas and now supply these organs as needed. Some mesenteries have specific names, and that's what we're going to talk about now. So the first is the lesser omentum, which I'm just going to call LO. And then we have the greater omentum, which I'm going to call GO. All right. And the lesser omentum is this small little part here that's going from the liver to the stomach. And so we will just put it in right here, lesser omentum. And the greater omentum is this big flap, and it ends up being a 4-layered fold because it doubles back on itself. See over here where my cursor is moving? And it is in front of the stomach and hangs kind of like a fatty apron over the stomach. And this is the greater omentum. All right. So that is in that location. Now, there's two others that I really want to talk about really quickly, and one is mesocolon. I might be able to type in mesocolon right here, mesocolon, and you can see it is, I'll move this up so you can figure out where it is. There it is, right here, and that's holding on to the large intestine. So we have the lesser omentum, the greater omentum, the mesocolon, and the falciform ligament. So the mesocolon holds the large intestine, especially the transverse colon, the part that goes across the body, is being held by the mesocolon, and then the falciform ligament. And the falciform ligament attaches the liver to the anterior part of the wall and the diaphragm, and that's right here. I'll make it up. Oh, this one's not perfect. It's this little, I guess we'll have to do it here, falciform ligament, FL. It's this thing, this little tiny thing right there. That is the falciform ligament. Okay. So it's good to know some of these. We talk about them occasionally. We can't, but I mean, I'm not going to go into a lot of detail about them, but there they are. It's good to kind of just do a quick overview of those different landmarks there. Okay. This is an abnormal condition about increasing fluid. And I'm going to skip for now. And then I want to talk about retroperitoneal organs. Now, retroperitoneal organs are organs that start in the peritoneal cavity, but then they eventually go to the back wall, and then they stay there. And so you can see here, and I'm going to draw arrows down because again I can do that. Right here, you see there's an organ in B that is out in the peritoneal cavity, but over time it attaches and goes into the back wall, and that's where it stays. And there's a lot of different organs which are retroperitoneal. So think of what that means. Peritoneal, it starts in the peritoneum and then it goes retro. It goes back. We think of retro as like back in time. Well, in this case, it goes back to the back posterior wall. So our kidneys are this way. They're embedded in the back wall of our peritoneal cavity. Our urinary bladder, our parts of our colon, parts of our pancreas, things like that. So that is a retroperitoneal organ. These here are retroperitoneal organs. These are typical organs that are just peritoneal. Okay. Now, next, what we want to talk about is splanchnic circulation. And this is just we want to talk about some of the circulatory pathways. And some of this we did in 232, so it's kind of a reminder of what we did before. So let's see what you remember. So splanchnic circulation is, and I'm going to kind of draw a little arrow here, all the arteries that branch off the abdominal aorta from the hepatic portal system. And so that is these guys right here. And so you might remember the celiac trunk, the first big branch off the abdominal aorta. That's right here. I'll type it in. Okay. And there are three branches. You have number one. Let's see if I can put it in right here. Number one. Right there, our common hepatic, for it goes to the liver. And then we have above it here, number two, all right, which is our left gastric that goes to the stomach. And then we have number three, and number three is our splenic artery, and our splenic artery goes to the spleen. So those are all listed right there. This diagram, by the way, is on page 744. Page 744, and this is back in our cardiovascular system blood vessels section. Okay? Now there's also the superior mesenteric and I put an extra S in, and the inferior mesenteric. Now, if you remember, these are going to supply small and large intestines, different sections of them, right? So a little bit of a review there. Then we also have the venous system, and that's our hepatic portal system. Now, the hepatic portal system, if you recall, a portal system means that we go from veins to capillaries to veins. And that's what we're seeing here. We start here, and these are all the nutrient-rich blood that we're getting from our digestive tract. So everything that we eat is going and being absorbed in our small intestine, and then it's going into these capillaries, and those capillaries eventually go into the superior and inferior mesenteric veins. Okay, and so those are right here, and I'm going to actually, just because I don't have a lot of space here, write superior for that one and write inferior for this one. And so mainly just so you can see, this inferior mesenteric vein is taking part of the digestive tract. The superior mesenteric vein is getting the blood from the rest. And this blood is rich in nutrients that we just absorbed. Then it goes to the hepatic portal system. And the hepatic portal system then will take all of that new food and nutrients that we just got into our body, and it will take it into the liver. And this is the liver right here, this big triangular thing. And then we can start processing it. If we have a lot of sugar, maybe we'll start storing some as glycogen. If we have some drugs or alcohol or something in there, we might start breaking it down and processing it. So we can do different things as it goes through the liver before it gets there. So those are just some reminders of things that you should kind of have remembered, but maybe not. All right. Now we're going to go back to the digestive system chapter, and we're going to look here at a couple. We're going to look at a typical wall of the alimentary canal. We're not looking at a specific one. I mean, like, we're not looking at, oh, this is the small intestine, or oh, this is the stomach. We're just going to look at kind of an average one, and we're going to see what structures do we normally find there. And I'm pulling up the page here while we talk, and I'm having trouble finding it. Let me see where it is. It's on page 873. Page 873. Okay. Now, I don't know if I want to draw in all the little things that are going to go right here. It'll be a little hard to do. You could do that yourself if you're doing your own paper. What I think I'm going to do instead is I'm going to switch over to the book, and I'm going to show you show it there, and then we'll go switch between the book and the lecture notes. Okay. Okay, so I just brought up the picture from your book, and this is the basic structure of the alimentary canal. And so what we can do is we can kind of go back and forth between the notes and the book here. So I'm going to zoom in just a little bit more so we can see those words better. All right, now we're going to switch back over. So the first thing that we're going to look at is the mucosa. We're going to start on the inside of this kind of typical GI tract wall. We're going to go out. So we're going to start the mucosa, the mucous membrane. And mucosa has an epithelial layer, the layer that's open to the lumen, the lamina propria underneath, and the muscularis mucosa layer. Okay, so what do those three things do? Well, you should know some of these already. The epithelium, remember, is going to be in contact with GI contents because it's the thing that lines the lumen. In the mouth and the esophagus, the epithelium is and the anus is stratified squamous epithelium. Okay, so for abrasion purposes. But the remainder of the tract is simple columnar, and this is better because we need to digest food in this area, so we don't want stratified epithelium. We want one layer of epithelium. Okay, and so the lamina propria is areolar connective tissue. And if you remember areolar connective tissue, it has all the different connective tissue types. It kind of supports and binds things together. And it's very vascular. It has lymph and blood. It has diffuse lymph tissue, some of which you'll see in lab slides, and other small glands. Some of those you'll see. This layer supports the epithelium, and it provides a blood supply, because remember, epithelium is avascular. The third one is the muscularis mucosa. These are smooth muscle layers, and they throw the membrane into many folds when they're contracting and relaxing, and this increases surface area. And we like increased surface area because increased surface area means that we can absorb nutrients better. There's a specific, or I'll give you an example of one type, which is the rugae of the stomach. That's how you spell it, rugae in the stomach. And these make the mucosa and submucosa wrinkle up. So when we see the stomach a little bit later, you'll see that kind of wrinkly inside, and that's coming from this muscularis mucosa. All right, now let's look at that. So where are we talking about here? Well, we just did all of these right here. The epithelium, the lamina propria, and the muscularis mucosa all together as one right there. Okay? Now we're going to turn the page, and we're going to look at the submucosa, which is this middle intermediate layer below the mucosa. Okay? So Submucosa is also areolar connective tissue, and it has a intrinsic nerve supply, a submucosal plexus, another one of these ANS nerve supplies, and it regulates the mucosa and glandular secretions. It's really vascular, the submucosa, and this is where we carry away our absorbed nutrients that go into our digestive system, and it also binds the mucosa to the next layer. All right, and that next layer is the muscularis layer or muscularis external layer. So let's look at our picture. So the muscularis external layer are here, and there's often multiple layers, sometimes 2, sometimes 3 layers of muscle tissue. And this is what's really doing most of the contraction and peristalsis or segmentation, whatever's happening is happening here. Okay? And then on the outermost layer is the serosa, right? The serosa is always the outermost layer. So we're going to talk about those two next. So the muscularis externa is skeletal muscle in some places. So places where you have control, like your mouth, your pharynx, and the top of your esophagus, it's skeletal muscle. So you can help initiate swallowing and move food in those areas. The remainder of the tract is smooth muscle, okay? And it's generally 2 sheets, circular fibers and longitudinal fibers. In certain organs, like stomach, there's 3 sheets there. In this layer, this muscularis layer, in this layer, the myenteric plexus exists, and this contains fibers from both parasympathetic and sympathetic. And so this is going to increase the MY is, you're thinking muscle, myenteric plexus. The parasympathetic increases motility, sympathetic decreases motility. Does that make sense? Remember that when we're in a sympathetic state, we usually quiet our digestive system. We don't want to be digesting things while we're escaping from a lion on the Serengeti plain or being chased by zombies or something. But when we're resting, relaxing, we want to increase our stomach, small intestine, large intestine motility and move and digest food through. So remember that. It's kind of the opposite of other parts of your body. The last thing we're going to talk about today, we're going to stop right here and do functional anatomy next time, is the serosa. Serosa is the outermost layer. This is the visceral peritoneum, right? This is the layer on the outside of the organs. So that's good to remember that now we're on the visceral peritoneum. And there is a plexus here as well. But I'm not as worried about it because it's not as important of the plexus than the other ones. However, in the esophagus, this layer of connective tissue on the outside merges with surrounding structures. And when that happens, we give it a specific name. And we call it adventitia. All right, so adventitia is when we have serosa that has attached to surrounding structures. And you're going to see that not only in the esophagus, but in a few other places where a tube or an organ needs to be attached to other areas. Okay? And so I think we're done there with a lot of the anatomy of the GI tract. So what we're going to do next time is we're going to start on the functional anatomy and go into specific parts of the digestive tract and how they work. Okay, great. We will see you next time.